13.3 is going to start out with just basic gene mutations, and this is any change in the DNA sequence, specifically more in DNA, but it can also happen in RNA. But we're changing some type of nucleotide sequence in the DNA gene that's going to regulate, or it's going to change the protein or protein expression in some sort. So what this is basically saying is some nucleotides change. An adenine is going to be changed to a thymine. You might insert or delete something. There's going to be some change in that nucleotide sequence. Now, usually those only affect an individual if they're in a germline cell, so like in the sperm or egg cells. So like body cells, they're not going to actually have any influence on your. So if you have a like a mutation in your skin cells, your son or daughter won't have that mutation. It has to be in the sperm or the egg. Now, mutations, they are very rare, and usually you have enzymes like DNA polymerase that's going to check for the like the mismatch of a protein, or I'm sorry, the mismatch of a nucleotide. And what happens is it can cut out that section of DNA and replace it. However, it's only 50% of the time efficient because it could be like the T could be the wrong or G could be wrong. We don't know which one, and the proteins aren't going to know. So this is how, even though proofreading and DNA polymerase is pretty accurate, it's not perfect. So mutations, a point mutation is a single change in the DNA. So again, one letter, you can see here, this one letter changes the DNA. This is one single nucleotide, and it can either be one of three or several others we're going to talk about here in a second, changes. So again, just one nucleotide. There can be a substitution where you change like an A for a T for a C, a G for a C, something along those lines where you're substituting. An insertion is when you input one nucleotide. So you can insert an adenine, you can insert a thymine, or you can insert a cytosine. And you can insert more than one, but specifically with point mutations, you only insert one. And deletion is exactly what it sounds like. You just remove one or several. Now, a missense mutation. This occurs when the mutation changes the amino acid. So sometimes you won't have an amino acid change if you, especially if you change that last nucleotide. Remember the wobble hypothesis. It won't change the amino acid sometimes. However, sometimes it does, and you will get a different type of amino acid. So you can see. Remember, if we back to chapter 3, we talked about how the amino acids have different properties. If you change the amino acid and it has basically the same property, that's called a conservative mutation. If you change the property totally, it's called a non-conservative. So this is like if both are acidic, lysine and arginine, and this is basic, or this is polar and this is non-polar. If it's the same property and it works in the same way and kind of folds the protein the same way, it's called a conservative. If it's a different property and it changes the form of the protein, it's called a non-conservative. Remember those. We have, again, acidic, basic, neutral, and polar, non-polar kind of amino acids. If you change like this amino acid to this one or this to this, it's a non-conservative. But if you change like these two, they have the same, or they have similar properties, so it's going to be a conservative. A nonsense mutation is a lot worse. It's a more, I should say, actually worse. It's a more, it affects the protein more because you change a amino acid to a stop codon, and that changes the protein drastically more because you might lose a lot of the end of that protein, which again changes the shape. When you change the shape, you change the function. So again, this is where you could have a protein that's like 1,000 long, 1,000 amino acids long, and halfway through there's now a stop codon. You don't get that second half of the protein. So imagine reading through a book and having halfway through the book, there's just it's just done. It changes the whole protein, or changes the whole story a lot. Silent mutations, again, these are really easy to remember because silent mutations is kind of what it sounds like. It doesn't do anything. You can see here where the mutation here, you still code for tryptophan, so it really doesn't matter, and it doesn't change the not the nucleotide sequence. It doesn't change the amino acid sequence. Frame shift mutations is one of the most detrimental. It's along the lines of the nonsense where you have that stop, which there's going to be drastic changes there. Not in the same case though. Whenever you have a frame shift mutation, you're either inserting or deleting a nucleotide, and you can see here just by changing the or adding a nucleotide here, we're pushing everything to the right one, and you can see those codons are now totally different, which creates a totally different sequence of amino acids, which totally changes the structure and function of that protein. And this can be either a insertion or a deletion. So this is a very drastic mutation. I like showing this as a example of the mutation. You can see the boy cut his lip and ate the hot dog. In a substitution, the boy cut his fip and ate the hot dog. You mostly kind of get the sentence. You can kind of guess what it means, and there's not much change there. But if you insert something, you can see here the boy cut his sla pa da. Sounds like you're almost speaking German here. It totally changes the sentence. And the same here with deletion. The boy cut his lip. Anna tet it totally again changes the sequence of or the letters and the word, so it totally changes. So substitutions aren't that I'd say drastic in changing the protein, but insertions and deletions are. Now, mutations. What happens is if even if there is a single mutation or a just one mutation, a change in amino acid, the protein can totally change, and it can either inactivate the protein, it can even speed up the protein. It can make the protein more efficient or less efficient. It's not always harmful. So a lot of times it is, but it's not always harmful. Sometimes it can actually be beneficial. And we're going to talk about mutations in evolution in the next couple chapters, but don't think that mutations are always bad. Sometimes mutations are really good. And I'm going to put this in the back of your brain now. You are actually just a bunch of mutations. Every gene is a mutation on itself, so it's not a bad thing. But a lot of times it's given that context that's bad, and it's not always bad. Just like I said here, we're going to talk about mutations and how, like normal hemoglobin, you have this GAA, but in a person with sickle cell, they have GUA, which changes a glutamine to a valine, and that creates that hemoglobin, that sickle cell shape. So just one nucleotide off can create drastic changes in a person. So we're going to see these mutations really take effect when we talk about evolution.